Right, everyone. Good afternoon. It's really a pleasure to welcome you to the Chemistry of Life Processes Institute, Feinberg School of Medicine, and Lurie Cancer Center Convergence workshop. Neil Kelleher was really excited to be here with us, but unfortunately, he's trying to make it back home. He's stranded right now in Las Vegas Airport. So I'm stepping in and doing the honors for today. It's a pleasure to welcome you all to this workshop. The theme for today is revolving around cancer epigenetics and also redox processes, including, of course, how oxidation of structural components of chromatin might regulate gene expression. And we're going to hear talks about how this could be a new connection between the metabolism, particularly the declining or the ever-changing metabolism of the cancer cells, and novel ways of gene regulation. Mechanisms of gene regulation. The goals of the workshop today are, of course, to introduce both clinical needs and also opportunities that the downtown clinical campus might have, and explore how the exceptional science that is done in biophysics and biochemistry, structural biology in the Evanston campus can help us to respond to those needs. Also, we aim at sharing unique technologies that exist on both campuses to see how we can synergize and establish new directions of research that might be useful to develop new therapies and understand the disease of cancer better. And of course, the ultimate goal would be to foster new interdisciplinary collaborations between the campuses and integrate these different groups into finding avenues for team science that can lead to large grant applications. Before we get started with the program, I'd like to share the program with you and go through some housekeeping items. First of all, the program will include 5 faculty presenters who will talk briefly about their research programs, about 10 minutes. We are going to have an extended Q&A session after that. And of course, all of the speakers will be available to answer your questions. Please make sure that you type your questions into the chat box at any given point, so we have a record of your questions and we can actually make them at the end. And during the Q&A session, you're encouraged to unmute yourself and ask your questions to the speaker of your choice. I will be serving today as the moderator and the presenter of symposium due to Neil not being able to be with us. The speakers are, well, I'm going to start. I'm going to give the first talk about our research in histone oxidation and chromatin remodeling, followed by Dan Foltz. He's a professor here in the Department of Biochemistry and Molecular Genetics. He also works on histone modifications and gene expression regulation. Yuan from the Evanston campus, who's going to talk to us about cryo-EM and some exceptional work that he has done in structural biology. Laura Lackner, working on mitochondria and its contacts with different organelles, exchange of metabolites between these organelles. And of course, John Marco, who's a professor of molecular biosciences, astronomy, and physics, who does not need to be introduced to this audience, working on the mechanics of the nucleus and how that can impact chromatin structure and gene expression regulation. So without further ado, I would like to start our symposium to kickstart. It's truly an honor to be able to give this talk about this new idea that we entitle Chicago Coalition for Cancer Genomics, Redox, Epigenetics, and Transcription, that aims at studying how changes of the electrochemical environment of the nucleus might affect different proteins, different structural components of chromatin, leading to new ways of gene expression regulation, particularly important for cancer cell biology and the evolution of cancer towards metastatic incurable disease. I am a professor of medicine here at Feinberg School of Medicine at the downtown campus, and also I am affiliated with the Department of Biochemistry and Molecular Genetics, and currently I serve as the Associate Director for Education and Training at the Lurie Cancer Center. So I have been trained as a biophysicist, and I have had a not so long career. I think we have been studying biochemical oxidations for the past 15 years. As you guys know, reactions and molecular interactions happen in an environment. And I think that my group has been thinking a lot about how biophysical forces that compose these environments where molecules exist and interact change their shapes and modify the efficiencies of these interactions. We are particularly interested in redox and processes because, most of the biophysical parameters, most of the biophysical forces that exist, such as temperature and pH, they don't change much. And if they change much, probably the organism is no longer viable. Little changes of decimals of pH values or degrees in temperature may lead to the death of most of pluricellular organisms, particularly more complex organisms. But with redox forces, the change in the concentration, minute changes, hyperlocalized minute changes in the concentrations of these species, because they are so short-lived and they exist in a very small volume, can lead to orders of magnitude differences in microenvironments or nanoenvironments being produced within organelles, within cells. And they might be very important to regulate processes by changing the shapes, just like temperature and pH do, of these molecules. Some of these reactions are reversible. They have been studied in the cytosol for many, many years. And we decided then to look at how the redox changes of the nucleus, of the nuclear envelope, might be contributing to oxidation of these molecules, changes in shape that might lead either to loss of epigenetic function or transcriptional function, gain of function, and what my laboratory is much more interested in, changing function. So to be able to study how these the changes in the reactive oxygen species or in the electrochemical state of the nucleus could be affecting cellular behavior or cancer cell behavior, we first had to come up with a model of how we could generate tunable amounts of oxidants in the nucleus, and we generated this new nuclear localized D-amino acid oxidase system. So what it does is that when you feed externally D-amino acids, remember that most proteins and most peptides in your body are actually made of L-amino acids. When we feed the enantiomer, D-amino acid oxidase, which is a fungal protein and yeast protein, now converts this D-amino acid into hydrogen peroxide that is locally generated. And we sent it to the nucleus by fusing this construct with the CMYC nuclear localization sequence. When we produced reactive oxygen species in the nucleus, hydrogen peroxide, and subjected samples to EM to look at the nucleus, we saw something very, very interesting. These are MCF10A cells. And you see that when they are under oxidizing conditions, when hydrogen peroxide is produced in the nucleus, we see the dissolution of heterochromatin, these denser regions of chromatin that accumulate at the inner side of the nuclear envelope. And we see an expansion of the size of the nucleus, probably to accommodate this more expensive chromatin. We started to think what could be happening here, why this could happen. And we realized that among the oxidizable components of chromatin, there is a histone 3 variant, histone 3.1, that contains a thiol residue and could be susceptible to oxidation. So when we mutated that residue, you see that there's no loss of heterochromatin anymore, and of course, this mutant is resistant to oxidation. We also see that there is no change or appreciable change in the volume of the nucleus, indicating that histone 3.1 that marks heterochromatin is an oxidizable component of chromatin, and oxidation of this histone might be important for chromatin organization. Showing to you here, if we actually produce reactive oxygen species in the nucleus, this variant 3.1 is degraded with hydrogen peroxide, but if that residue is replaced by oxidation-resistant amino acid, in this case serine, you don't see the loss of 3.1 anymore. I don't have time to go through a lot of detail in our research, but one thing that we discovered is that the accumulation of reactive oxygen species in the nucleus is very important for how the tumor cells respond to chemotherapy because of this effect on histone 3.1. What you see here is PY230 mouse carcinoma cells. They have, as most cells, a lower level of reactive oxygen species in the nucleus at baseline. When we start to treat these cells with chemotherapy, in this case, we use doxorubicin, but it's true for a lot of other kinds of chemotherapy. You see that the vast majority of the cells die, but the residual population that you have here has both accumulated reactive oxygen species, probably because chemotherapy damages mitochondria, increases reactive oxygen species production, and that elevates these species in the nucleus, leading to cell plasticity and the capacity to adapt to the drug treatment. If after a few days, these high nuclear ROS cells start to proliferate in the presence of chemotherapy, but if we activate catalase in the nucleus, catalase degrades hydrogen peroxide, removes hydrogen peroxide, we now resensitize these cells to chemotherapy, to doxorubicin in this case. And that really closely parallels the accumulation or the stability of histone 3.1 in the nucleus. You see that very quickly after we start treatment, we lose histone 3.1. Chromatin gets more expensive. Cells can explore a larger range of transcriptomes to adapt to chemotherapy. When we turn on catalase, 3.1 returns, and that leads to the resensitization of the cells to chemotherapy. So the question is, are there subsets of genes that are harbored in heterochromatin that are responsive to redox regulation and involved in this adaptive process to treatments, to stress that cells undergo and cancer cells undergo? And it was a very fortunate time moving here to Northwestern. I actually got to meet Vadim Bachman, and as you guys know, he studies mechanism of chemoresistance activation by cancer cells and how chromatin folding heterogeneity contributes to that. This is one of his beautiful images where he looks at chemotherapy, and chemotherapy increases chromatin folding heterogeneity. That leads to chemoresistance, and I started to hypothesize that maybe changes in the redox state of the nucleus could underlie chromatin folding heterogenization and contribute to what he's seen from his side. And this is just a first experiment where we actually produced reactive oxygen species in the nucleus with our nuclear D-alanine oxidase system. We could measure with our biosensors, and I'll be more than glad to discuss how it works with you guys later. And then we did a PWS and MSA analysis to look at the transcriptomic landscape of the cells. And sure enough, increasing reactive oxygen species in the nucleus much amplifies both chromatin folding heterogeneity and the variety of transcriptomes, the transcriptomic landscape that the cells can explore. So establishing a direct connection between our groups that led to the thinking of a more expensive project that involves a lot of us here in the workshop today and others that we wanted to call C3 Great, or the study of the redox epigenome as a new target for cancer treatment. And we really have three aims here. And of course, these aims are very nascent. They can be modified. Others are very welcome to participate in this effort. So just talking about aims, but these are larger projects. We wanted to understand and develop an atlas of oxidizable structural components of chromatin and determine how they impact gene expression. Of course, that evolves from our work with histone 3.1 as a first example of these structural components that can be oxidized. We wanted to determine how these redox-driven changes to chromatin architecture affect domain organization, histone utilization, distribution, and how oxidation affects other important post-translational modifications, methylations, acetylations, and etc. And of course, we would like to collaborate with our colleagues and friends that are more interested in cell metabolism and understanding of what are the sources of reactive oxygen species that feed this ROS to the nucleus, how the context between these organelles and the positioning of these organelles close or further from the nucleus enable gene expression via this mechanism. So that's what C3 Great wants to accomplish. And that's what I would like to bring as an introduction to you today. I think that we are going to reserve the questions for the end of the talks, but thank you very much for being here, and thank you very much for your attention. So now I think I will introduce Dan, and then I have you to share your screen. Thank you. And now I'm going to go for a...
All right. So good afternoon, everyone. My name is Dan Foltz. I'm in the biochemistry department on the Feinberg campus. And I appreciate the opportunity that Neil has given us here today to get together and talk chromatin, and to Marcelo for really introducing some potential new overlaps. So I'm going to tell you two stories that we're working on in the lab right now, that some of which have metabolism bents and some of which do not, but nonetheless we're interested in seeing how these different domains do meet in the lab. So my lab has historically been interested in understanding chromosome segregation. And the reason that we've been interested in this is because most tumors are in some way aneuploid and demonstrate some level of chromosome missegregation and genome instability. This is a classic picture of a Sky karyotype. On the left is a normal cell, and on the right is a cancer cell, and it's rather obvious the large degree of genomic instability that many of these cells have. You can see that there's different types of chromosome instability. There's numerical changes in the karyotype, which you can see by a number of these purple chromosomes. But you can also see that there are many rearrangements and structural changes to the chromosomes. And so in the context of numerical changes in the chromosomes, this is oftentimes due to failures in mitosis, specifically in failures of separating the cells, but also in segregating of the chromosomes during mitosis. But it turns out that errors in chromosome missegregation can also lead to structural changes in the chromosomes through processes such as chromothripsis and others that have been identified. And so we've been interested, really in the mechanics of chromosome missegregation, really from the context of chromatin assembly. And that's because I'll point out here is that centromeres, which is the red dot shown on the chromosome here, are really specified by the presence of a unique histone that gets deposited into that site by a unique chaperone called HJURP. And that's really the definition of a centromere. And that's what selects one region along a chromosome to become a centromere. And that process is important for maintaining chromosome stability. If you have multiple centromeres along a chromosome, of course, you can get attachment to both poles and cause chromosome missegregation. And that can lead to chromosome breakage, fusion, bridge cycles that are also origins of structural rearrangements. But the general concept that I want to get across as well through this slide and what we're interested in is not only how centromeres utilize a unique chaperone, but the fact that histone variants in general, which serve different functions within the cell, utilize different chaperone processes in order for their unique temporal and spatial distribution within the chromatin. And so I'll tell you two short stories today. One about how we're looking at chromosome and centromere dysregulation in cancer. And the second, how we think we've uncovered coordination between nucleotide metabolism and histone chaperone pathways. So I'll first just highlight that while we've been interested in the centromeric nucleosome, that centromeric nucleosome then goes on to build quite a large structure that mediates the interaction of the chromosomes with the microtubules. And that the first layer of that is this CCAN complex, which are a set of proteins that are present at the centromere throughout the cell cycle. And we know quite a bit about how canonical centromeres are now deposited, in the context of normal chromosome missegregation. But we're very interested in understanding how those processes are perturbed in cancer. And so if we look across different cancers, this is just data from TCGA, we see that there's upregulation of a number of the regulators of centromere and kinetochore gene expression, which is the FOXM1 and the MIB-like-2 transcription factors, as well as the SEMP-F protein. And this is a project that's really being driven by Sakshi Khurana in the lab. And what we've seen is that most of this CCAN complex is upregulated in the context of cancer, including that SEMP-A histone variant SEMP-A, which makes the centromeric nucleosome. The exception is this SEMP-C protein, which is an interesting story that that I'm not going to really talk about today, but is a linchpin in connecting the centromeric nucleosomes to the kinetochore. But what we've been trying to do is understand what SENPE overexpression really means to the cell and what the ultimate phenotypes are of having too much SENPE in the chromatin. And one possibility is that that I won't talk about today is that the presence of extra SENPE really decreases the function of the endogenous centromeres, and we know that's true for a number of examples. But what we're also seeing is that we think that overexpression of SENPE is leading to the assembly of centromeric sites outside of the canonical centromere, and also that SENPE being incorporated outside of the centromere also leads to dysfunction of that underlying chromatin. And we're doing this by using a SENPE inducible system where we're now overexpressing SENPE. This is a cut and run experiment. Here we see that we've induced SENPE with doxycycline, and you can see that under normal conditions, there's very little SENPE except at the centromeres. And now we've kind of littered the SENPE throughout the arms of the chromosome. And this is true in all chromosomes. I'm just showing you chromosome 2 here as an example. What happens is that SENPE then gets deposited in sites that it doesn't usually exist at, and we find that CTCF sites are actually hotspots for centromere deposition, proposing the idea that perhaps 3D genome organization is disrupted by SENPE overexpression. And the other thing that we find interesting in these experiments is the fact that if you look at accessibility of the chromatin, most sites, even though they have SENPE deposited at them, don't really change in their accessibility, but a subset of sites are either lose or gain accessibility in those sites. And we think that those sites, the sites that are gaining in accessibility, are actually forming neocentromeres, because that's exactly what we see at centromeres. Is with the presence of SENPE, we now gain accessibility within those regions. And so Pranati Vadlamani in the lab is really working to understand how those sites may be different. But in addition, we've also observed that SENPE being deposited into the non-centromeric sites along the chromatin also leads to changes in the DNA damage response. So I'll remind you that the DNA damage response is mediated by phosphorylation of phospho-H2AX. And so if we upregulate SENPE in these cells and then challenge the cells with some sort of DNA damage, we see that cells that have overexpressing SENPE fail to upregulate that DNA damage response, as indicated by this phospho-H2AX staining. And the quantitation is just shown here on the right. And this is consistent with what others have seen in the literature, that SENPE overexpression leads to actually a desensitization to some of the DNA damaging agents, suggesting that the cells don't have a good response to the DNA damage, although the underlying DNA damage is occurring. And so Alex Lee has taken an approach in the lab to be able to biotinylate all the proteins that are associated with centromeric nucleosomes using this turbo ID system. And what he's found is that when he looks at SENPE relative to other canonical histones, he sees that phospho-H2AX component of the nucleosome is absent. And so what we think is that the nucleosomes that are deposited along the outside of the centromeric regions are actually refractory to some of the responses to DNA damage that cells would normally have, thus blunting the response that we're seeing, and potentially increasing the amount of persistent DNA damage that we'll see in these cells. And then the last couple of minutes of the slide, of the talk, I'll just tell you a little bit about this canonical chromatin assembly and the connection to nucleotide metabolism. And this is a project that's been spearheaded by Shashank Shrivastava in the lab, who's a really excellent postdoctoral fellow and presently going on the job market. And so what I didn't tell you is that the chaperone pathway is a little more complex than I had initially intimated, and there's quite a few different chaperones that are involved in different components and different parts of facilitating movement of histones from synthesis to deposition in the chromatin. But what I want to highlight is kind of these early steps. And a little bit earlier in our work, we identified a protein called UBR7 as being a canonical histone chaperone that seems to interact with one of these components in this pathway called NASP, which is very early in the process of nucleosome chaperone and deposition. And so Ann Hogan in my lab identified this as a histone chaperone, but when Shashank started looking at this protein a little bit more closely, he identified some interesting connections to the nucleotide metabolism pathway. And the first thing he observed is that UBR7 is highly upregulated in T-cell acute lymphoblastic leukemia. And when he suppresses UBR7, he can actually suppress the TLL in xenograft experiments. And that was because it turned out that UBR7 actually bound a set of proteins called PRPS1 and PRPS2, which are rate-limiting enzymes in the production of nucleotide metabolism. And he could show that when he suppresses UBR7, he actually suppresses PRPS1 activity and reduces nucleotide metabolism. And through a number of experiments showed that this is dependent on a ubiquitination of a negative regulator. But what really piqued our interest, being actually more of a chromatin assembly and a chromatin lab rather than metabolism lab, is that if we affinity purified UBR7, which we had shown is a histone chaperone, or if we purified another histone chaperone called NASP, in both cases we identify these PRPS enzymes, which are these rate-limiting enzymes in nucleotide metabolism. And so Shashank has been asking whether there's an effect of PRPS on the chaperone pathway for histones. And so he's done that in a couple of different ways. He's, of course, knocked down PRPS1 using shRNA approaches, or PRPS2, which is a closely related ortholog. And in both cases, what he can see is the accumulation of histones and the accumulation of specifically new pools of histones, which we identify by the acetylation mark on H4K5 and H4K12. Using the Kelleher and the Core approach to profile histones within the chromatin, we can see that those new histone marks are actually lost from the chromatin, suggesting that new histones aren't actually making their way into the chromatin. Shashank has then expanded this using the DTAG system and sees that this happens with fairly fast kinetics within one cell cycle of degrading PRPS1. And then when he degrades PRPS1, what happens is that the histones seem to be accumulating in this very early stage of assembly associated with this HSP90 component. And so what we think is happening is PRPS is a required component of this chaperone pathway, and that in the absence of PRPS, that the histones actually remain bound to NASP and HSP90 and inhibits their ability to progress into the assembly pathways. And now we're trying to understand two things: one, how the enzymatic activity of PRPS and the production of nucleotide, which is required kind of at the same time as new histone synthesis in preparation for S phase, are somehow coordinated through this pathway. And ultimately, what the phenotype is of inhibiting PRPS1 on the deposition and distribution of histones within the chromatin. And with that, I will stop and thank specifically Shashank, who's postdoc in the lab, and Alex, as well as Pranathi, who have been focused on understanding SEMPE overexpression. And thank you for your attention.
Thank you so much, then. Beautiful presentation. The next speaker is Yuan. Hey, Yuan, great to have you here. So Yuan is a professor at the Department of Molecular Biosciences. His research is heavily focused on mechanisms that regulating gene transcription, DNA repair. He has had some very interesting publications recently, very high impact ones using cryo-EM to dissect mechanisms of gene transcription regulation, depending on timing and phase, and also complexes of chromatin remodeling complexes, protein complexes, and nucleosomes. Looking forward to your talk, Yuan. Thank you.
All right. Thank you for the organizers to give me the opportunity to talk about our research. My lab is interested in using a combination of approach, including biophysical, biochemical approaches to understand how eukaryotic cells read and repair our genome. A major biophysical tool that we use is a single particle cryo-EM, and I'm going to give you just a quick tool of the technique and some recent progress that we use this technique to visualize chromatin regulatory complexes. So as you probably know, in 2017, the Nobel Prize in Chemistry was awarded to 3 pioneers in the field of establishing this technique to really revolutionize structural biology, to allow us to use very limited amount of material, either roughly purified from cells, or in recent years, looking at many of these machineries inside of the cell toward atomic resolution. So the process starts with, in this case, purified proteins. Shown in this example is a GROW-EL, freely floating inside of the tube. The question is how to actually visualize in a TEM setup. So we use a copper grid, a 3 millimeter copper grid, apply 3 microliter samples to the grid in physiological relevant concentration buffer conditions, and then use a vitrobot or vitrification robot to blot majority of the sample away to leave a very thin layer of the sample containing those particles on the grid. And this process is maintained in 100% humidity, and the grid, along with the sample, can be frozen using liquid assay. So what I describe here is going from dynamic sample to a single layer, a thin layer of frozen hydrated material for visualization in the vacuum environment in TEM. This is an old picture. Now we have a glacios at the Evanston structural biology facility, allow us to image these particles in a more automatic manner. So the process of image data collection is electron is going to interact with these various particles, and we assume the particle are adopting in various different orientations, and the interaction of the electron beam with the sample will leave a shadow, a 2-dimensional image onto the camera, and that's basically your data. And we collect thousands of these micrographs, and each micrograph contains hundreds of these particles, and eventually you can get to millions of these particles. And the process of data processing starting with particle picking. So you pick the particle that adopt a very similar orientation. In these cases, you can actually see these are the same view, and then you can put them out into a stack, orient them, align them, and then the process, this process is fully automatic using clusters, and you can actually get a much crisper image of the GROW-EL in this orientation. And then you exhaust the view of all the micrograph you collected from different views, and in 3-dimensional manner, you can reconstruct all these 2-dimensional image into a 3-dimensional manner. And the movie was made over 15 years ago. So at the time, you can see these alpha helices appear as sausages as state art. Currently, you can actually see atoms. So far, the record is close, approaching 1 angstrom resolution. You start to see really individual atoms. So this is just quick a brief introduction of the technique. My lab is interested in mainly two fields. One is gene regulatory complexes. We're very interested in the basal machineries, including the eukaryotic RNA POL1, 2, and 3, illustrated here. They are involved in various process of transcription for production of either ribosomal RNA, tRNA, or messenger RNA. What we learned by comparing POL1 versus POL2, showing on the left and right respectively, that POL1 actually utilized its core factor to open the DNA bubble in a very different manner as what's shown on the right for POL2 system. In the case of POL2, you have a DNA helicase to facilitate the bubble opening process, whereas it relies on the intrinsic disorder flexibility of the generation factor involved in POL1 initiation to open the bubble up. By comparing with POL3 pre-initiation complex, we also conclude TF3B plays a role in facilitated recycling of POL3, probably by engaging POL3 throughout the cycle of transcription, whereas you can see even from this view, the comparison of POL1 look very differently from POL2 and POL3. My lab is also very interested, besides the basal machinery, we're also very interested in other co-regulator complexes, including ATP-dependent chromatin remodeling complexes, modifying complexes, mediator, and so on. I'm going to just give two examples here. First is we were able to capture for the first time the eukaryotic switch SNP chromatin remodeling complex in association with a mononucleosome and illustrate how it can actually couple the ATPase in the SNP2 subunit to the immobilization of the histone octamer to result in a natural sliding of the DNA relative to the histone octamer. And we were also able to in vitro reconstitute and capture a 57 subunit complex involving the human POL2 initiation complex with mediator bound. So we also learned how this mediator complex played two critical roles. One is to recruit polymerase to the promoter, where on the other hand, stabilize the CDK from TF2H to facilitate the phosphorylation of CTD of RPP1. Besides that, another half of my lab is also very interested in understanding the mechanism of how double-strand break is being detected and repaired. There are many complementary pathways involved in this process. Shown here are two major ones: recombination or non-Hodgkin end-joining. And we were also able to use single particle cryo-EM to visualize, to capture two critical states. The first one is a long-range complex where the kinase recognize the Ku70, Ku80 ring to bridge the two complex, two half of the double-strand break together to facilitate this transition into the short-range complex to allow the loading of the ligase catalytic domain to eventually repair a double-strand break in this manner. So with that, I would like to thank people who have contribute to the work I have described today. Ryan and Anna contribute to the mediator PIC work. Yan contributed to the POL1 and POL3 work, whereas Siyu Chen contributed to the NHEJ work. And thank you for your attention.
Well, thank you so much. Fascinating talk. I think that we're going to have to save the question for the end. Next speaker is Laura Lackner, who also comes from the Department of Molecular Biosciences. Laura is interested in how the organization of organelles within the cells and their context modulate their function, as well as cellular behavior. So thank you so much, Laura, for the presentation.
Okay, excellent. Thank you so much. Okay, sorry about that. Thank you so much for including me in this workshop. So I can say I don't directly work on redox epigenetics, but I do work on an organelle that is directly impacting redox epigenetics. And so in my lab, we're really interested in understanding the mechanisms that shape and position mitochondria. And so when I say mitochondria, oftentimes people immediately think powerhouse, the powerhouse of the cell. And I know this audience knows that mitochondria do more than produce ATP through oxidative phosphorylation. They have a myriad of functions in cells that are really critical for how cells function. These include things like metabolite exchange. They house apoptotic factors that need to be released during apoptosis. They're important in creating iron sulfur clusters, which are an essential cofactor for many enzymes. They're important for phospholipid biosynthesis. And relating to redox epigenetics, they're also really important for TCA cycle as well as ROS generation. And we know that both TCA cycle intermediates as well as mitochondrial generated ROS can play a role in redox epigenetic regulation. And so when we think about these many functions of mitochondria, we know that these functions can be influenced by mitochondrial form. So just like many things in biology, form can influence function. So when I talk about mitochondrial form, what do I mean? And that really depends on what cell we're looking at. And so depending on cell type, mitochondria can adopt a wide variety of shapes. So here are mitochondria in my favorite model system, the budding yeast Saccharomyces cerevisiae, where you see mitochondria adopt these beautiful reticular networks that are well distributed across the cell. These reticular networks are also seen in parasites such as toxoplasma. You can also see them in mouse fibroblasts. And I also like to highlight here the axon of hippocampal neuron as a contrast, where you can see in this region of the cell, mitochondria form the more typical bean-shaped structures. What we know in all cell types is mitochondria are dynamic. They're constantly moving and rearranging within the cell. And we know these dynamics are also used to then adapt mitochondrial shape and positioning to meet the needs of a cell. So if you look at kind of a cell in the mitochondrial shape it needs at a given moment, that can really depend on what it's experiencing at that time. So if you look at the budding yeast, once again in mitosis, these beautiful, well-distributed reticular networks. However, if you force the cells to undergo sporulation or meiosis, what you can see are dramatic changes in that mitochondrial network, where the mitochondrial network comes off the plasma membrane and has to be encapsulated into spores. If you look at fibroblasts that are treated with apoptotic stimuli, what you can see is that mitochondrial network fragments, which is thought to help release apoptotic factors. And another example is when we look at, say, migrating cells, whether they be cancer cells or other migratory cells, what you can see is that in the leading edge versus the trailing edge, mitochondria tend to be trafficked to that leading edge of the cell and kind of retracted from that trailing edge. So you can see, depending on what's going on in the cell, there has to be these dramatic rearrangements of those mitochondrial networks. And so how are these kind of rearrangements taking place? You know, what's regulating these restructurings? And so what we know is, when we think about it quite simply, if we think about mitochondrial shape and positioning, there are 4 simple activities that can regulate that in cells. And those activities are fusion, division, motility, and tethering. And so fusion and division regulate that interconnectivity of the mitochondrial network. Motility is important for trafficking mitochondria to specific cellular locations, while tethering is important for maintaining mitochondria at those locations. And so my lab and many others are really interested in the molecular mechanisms driving these processes of fission, fusion, motility, and tethering. But what we've begun to realize is that we can't just keep a mitochondrial centric view. We have to think about kind of the rest of the cytosol when we really want to think about the activities that shape and position mitochondria, because we know that the activities that shape and position mitochondria involve contact with other organelles. So we know that mitochondria pretty much make physical contact with every other membrane system in the cell. And these contacts have been shown to play critical roles in the activities that shape and position mitochondria. But we also know that these contacts mitochondria make with other organelles play many other functions, and the list of functions that are attributed to mitochondrial membrane contact sites is continuing to grow. And I just want to highlight some functions of this particular contact, the mitochondria in the nucleus, because it's most relevant to this workshop, where this physical contact between mitochondria and nucleus is thought to be important for nuclear mitochondrial communication, potentially lipid transport, as well as molecular transport. And so really we knew mitochondria made contact with organelles, especially the ER in the 1950s, but really the field has kind of been rejuvenated in the past 15 to 20 years. And really a lot of exciting things have been done in the field of mitochondrial contact site biology, but it's still kind of relatively young with what we're learning. And so there are some fundamental questions we can address. And these are questions that my lab is interested in answering. So what are the proteins involved in organelle tethering at contact sites, and how do they tether? How are contact site formation and function spatially and temporally regulated? How do contact sites influence overall mitochondrial function and overall cellular fitness? What are the additional functions of contact sites that have yet to be described? And then can we manipulate tethering proteins? So the proteins we identify that mediate tethering between organelles, can we manipulate them to control mitochondrial positioning and or contact site function? And so the last question is what I want to focus on for the rest of the talk, highlighting some kind of new tools and new ideas we have in the lab. And so the lab studies many membrane contact sites, but one contact site that has been a focus of the lab is between the mitochondrion and the plasma membrane, and this contact is mediated by a protein called NUM1. So NUM1 forms this contact between mitochondria and the plasma membrane, and this contact is truly multifunctional. So not only does it tether mitochondria to the plasma membrane, but we know this contact is important for mitochondrial distribution. It's important as a place where dynein gets anchored for its role in nuclear positioning. It's really important for maintaining mitochondrial function. We know that this contact is important in some way facilitates mitochondrial division. We have recent evidence that suggests this contact is also involved in lipid homeostasis. And we're always trying to search for new functions we can attribute to this contact site. And so we've spent a lot of years really kind of dissecting this contact, really understanding the protein, but kind of where we are now is we really want to begin to manipulate this contact site when we want to. And so we're beginning to ask questions of, can we control contact site formation and examine the downstream consequences in real time? And is simply tethering mitochondria to the plasma membrane sufficient to restore mitochondrial functions at a site of contact? So in order to begin to address these questions, we're building regulatable versions of a native contact site, as well as artificial mitochondrial plasma membrane tethers. And so what I want to highlight is some of these tools we're building and how we're using them to address kind of the role of this contact site in lipid homeostasis. And so what do I mean by this? So we recently discovered a role for this contact site in the distribution of plasma membrane PI4P. So PI4P is a phosphoinositide that plays many important roles at the plasma membrane. So in yeast, PI4P is primarily enriched at the plasma membrane of daughter cells and wild type cells. However, when we delete NUM1, this contact site, what we see is that more cells have PI4P in the mother cell. It loses this asymmetric distribution, and we can quantify this distribution here with this Bud enrichment score. So we were curious to know, could we actually, if we restored contact with the plasma membrane, rescue PI4P distribution? So to address this question, what we made use of is an inducible contact site that we built. So we know a lot about NUM1, and because we know a lot about its domain structure, we were able to introduce a rapamycin dimerization system in the middle of the protein. So in the presence of rapamycin, we're able to reconstitute that native membrane contact site. And this is shown here. So in the absence of rapamycin, we don't see the contact site. When we add rapamycin, what we begin to see are these contact sites form, and they're beginning to physically tether mitochondria to the cell cortex. And what's interesting is we can see this happen within 5 minutes. So within 5 minutes after rapamycin addition, we see about a wild type level of tethers form. We can then use these tools to begin to address questions about this contact site. For example, can we restore PI4P distribution in these cells? And what we see is that when we add rapamycin using our inducible tether system, we are able to restore PI4P distribution. However, while contact sites form within 5 minutes, PI4P restoration of distribution happens around 1 to 3 hours. So it's a much slower process. We can then use similar methods of these tethering systems to see if we require NUM1 specifically to tether mitochondria to the plasma membrane, or if artificial tethers could actually reconstitute the functions of this plasma membrane tether. And so what we've done is also built artificial mitochondrial plasma membrane tethers, where we take mitochondrial targeting domains of known proteins, and we use a tool that I'm happy to go into later, where we can then recruit these domains to the plasma membrane and restore this beautiful reticular distribution of mitochondria. And when we look at PI4P distribution in the absence of NUM1, what we see are these artificial tethers are not able to restore PI4P distribution. So I just wanted to highlight kind of like, once again, some of the tools we're building where we can manipulate mitochondrial positioning and really look at kind of the downstream consequences of forming a contact site, and really being able to determine if a protein at that contact site is important for the function, or is it just bringing two membranes into close proximity. So we're expanding these tools to other membrane structures. And so we're able to now target mitochondria to the vacuole, which is the yeast lysosome, as well as to the paranuclear ER, which would be important kind of for the subjects we're talking about here. And so that's kind of where I want to end, showing kind of those nice little vignettes. but I really want to thank the people that did the work, and that's Jason Kassler, Claire Harper, Heidi Anderson, and Antonine White. And I'm looking forward to the discussion at the end of the talks.
Thank you so much, Laura. Fascinating. I hope we'll have a lot of time. I have questions for every single of you already. Anyway, our last speaker of the day is John Marco, who is joining us from all the way across the Atlantic. So thank you very much for doing that, John. And John is also a professor at Department of Molecular Biosciences, also astronomy, and he is interested in applying methods and the discipline of polymer science into understanding how DNA and proteins interact, leading to chromosomal and chromatopolymization and gene expression.
All right, hold on a sec while I share my screen suitably. I'm here at a conference in Il Chioco that some of you guys might know. Some of you are going to Little Italy later. Well, I'm in Big Italy.
In Big Italy, jealous.
Just hold on a sec here. I'm sort of sitting here in the hotel lobby with dogs and cats, and where is he? Should be able to do this. Okay, all right, we'll do it this way. Share the whole dang screen. And then do this. You guys see my notes?
Slides look good.
Yes.
You're seeing something?
Yes. Thank you.
So I originally got this email from Marcelo, and I was very excited. And he said that he was interested in the possibility of starting a group focused on redox chemistry associated with gene expression and related to the impact of reactive oxygen species and other, I assume, redox processes on chromatin organization and all of the good stuff associated with that. And so what I wanted to talk to you guys today about is not to give you like a 5-minute elevator talk about what we're doing in our lab, but to talk about something that is a loose end that I think is related to this P01 research topic, and also to talk about what I would see as like a kind of a dream possibility for a project in that P01. So you guys are familiar with the dramatic changes that happen during the cell cycle to chromosomes, and they get refolded as metaphase chromosomes eventually. And there's a picture of a metaphase spread. And this picture was actually made by a former IBIS student named Adrienne Eastland. And she was carrying out a project to follow up on an observation that another student had made, a guy named Ryo Kawamura, that showed that, surprisingly, we discovered this really by accident, that when you expose metaphase chromosomes isolated from cells, we do mechanical experiments on them. That's, you can see over here, a chromosome on the right, suspended between two pipettes, and we can use those pipettes to measure the mechanics of the chromosome. Well, the mechanics of the chromosome, as isolated immediately from the cell, shows an elastic response that's hugely changed. In fact, it's made hugely weaker, as shown by the D curve, showing force versus extension of a chromosome. This in particular one is from an amphibian cell, but it works the same from human cells or mouse cells, after application of an antioxidant, in this case, DTT. Same thing happens with TCEP, BME, or reduced glutathione. And so this suggests that there may be some role for redox chemistry in modulating chromatin structure at a very basic level. So I was very excited by Marco's focus on this kind of thing connected with histone as well. So a thing we did that was a lot of fun is Adrienne developed a system for labeling oxidized cysteines on metaphase chromosomes. And so what you see here are some pictures that are black. Those correspond to these bars over here, which are at 0, for what you see after you block all oxidizable cysteines with maleimide and then try to label them with fluorescein malamide, or block with malamide and don't label with fluorescein, and you just see black. Now, if you take the metaphase chromosomes from a different collection and maleimide block them, and then reduce the reducible species that are left, which don't include all of those malamide sites you blocked, and then label, you get this box over here on the right, which shows some labeling. And in case you're wondering, is everything oxidized when you take it out of the cell? No way. If you don't block and you don't reduce and label with fluorescent malamide, you get this very bright signal on the left lower here. So most cysteines survive isolation from the cell and are not oxidized because they're in a neutral kind of environment outside the cell. But there are a lot of cysteines which, after blocking the free cysteines, can then be reduced. So what this means is that change in mechanics of the chromosome might be related to oxidized cysteines, which are actually functional in the oxidized state to hold the chromosome together. So what Adrian went on to do is to use the same kind of labeling technology to, on the left here, you see a SDS page gel of chromosomal proteins. You see a lot of histones at the bottom. And then these dark bands up here represent major chromosomal proteins, including topoisomerases and SMC complexes. And what she showed was, she was able to then, on the right is shown fluorescence in green, on the green panel is fluorescence, where you carry out this approach. The last band shows blocking of oxidizable cysteines, reduced cysteines with blocking reagent, in this case maleimide, and then reduction of any oxidizable, reducible species of oxidized cysteine with TCEP, and then labeling with fluorescein. And you see these dark bands here, which represent the species, some of the species, which are now labeled as being oxidized inside the cell, but not reduced inside the cell. So we did similar experiments instead of using fluorescein with N-ethylmalamide, which was suggested to us by Deval Nanavati, who was working at the Proteomics Center at the time and was a big help in this project. And that provided a mass label, which allowed us to show that these proteins were actually a large set of proteins, including cohesin, SMC1 and SMC3, major components of the cohesin complex, condensin 1 components, but no condensin 2 components, RCC1, RCC2, and P54NRB. Now, I have to tell you guys that there have been a few scattered reports of this kind of oxidized proteins playing a role in chromosome structure, but this is a kind of unmined area of chromatin structure. So if you look in the literature, you'll see this sporadic literature developing over many years, where not only are people talking about redox chemistry playing a role in chromosome structure, but also that there may be a redox cycle associated with the cell cycle itself. This is poorly established at this point, but I would point out that there's a brand new paper appearing in Nature Metabolism this month, like today or something, actually in a couple of days, which is on NADPH oscillations associated with mitotic progression in cancer cells. So this is something that some people are thinking about, and I think leads to kind of some major questions that would be perfect for the P01, I think that Marcelo is talking about. And I think it's still very poorly established if there are really redox oscillations during the cell cycle in normal cells, in cancer cells, in any kind of cell. And I think this relates to some of the kind of biology and biochemistry that Laura does, because she's concerned with mitochondria, which are a potential source of these kinds of variations. The thing that we don't know is what are the complete set of oxidized sites. In fact, the system that Deval developed for us based on trypsinized proteins, it was very clearly unable to resolve all of the sites on large proteins of size 1,000 or so amino acids. And that's for some, not very clear reasons. And I wonder if the proteomics state of the art has advanced in the last several years, which could allow us to do a more complete coverage of those large proteins like the SMC complexes. And if possible, I think probably people like Neil, it's too bad Neil can't be here today, might have some ideas about how to determine what the oxidized modifications actually are. We don't think, actually around our circle, we don't think that they're disulfides. It's not so simple. They're probably glutathionellations or other internal bonds which don't involve disulfides. We have our reasons for thinking that, but it would be great to have a chemical identification scheme for the oxidized modifications themselves. And of course, it would be great to determine the cell cycle variation of those modifications, and also to try to understand what molecular interactions, proteins and nucleic acids, proteins, the cell membrane or what have you, do those modifications regulate. And in short, my dream for this kind of project is to try to discover a cell cycle related redox code that would be a new level of regulation that I think has not been elucidated in the literature. So that's sort of my set of sub-aims for my P01 aim that I would be the head of. So that's my idea for a presentation here. That's it.
John, fascinating. Thank you so much. I'm gonna quote John Lennon here, right? A dream you dream alone is only a dream, but a dream that you dream together is reality.
Well, a dream plus a pile of money from the NIH becomes a reality, right?
Let's go get it.
All right, good. So no, we got one aim here ready to go. I could write an aim in like an hour.
Fantastic. It's a start. Thank you. I think that brings us to the Q&A session of the workshop. So I would like to invite everyone that is with us here to either type their questions. I can start. Actually, I have basically questions for all of you. I think I'll start with Laura. Laura, I would like you to expand a little bit. Of course, that tethering of mitochondria to different organelles will change function, but how established these are? Like when do we have some rules of thumb when it tethers with AR and calcium influx happens, what changes? Or when it tethers with the nucleus, do we have a catalog of these interactions and how it changes mitochondria, or it's still a very open area?
At this point, it's still a very open area because even different contacts, the mitochondria ER contacts are made by a whole range of different protein complexes. Even mitochondria nucleus now, which is becoming a much more interesting contact and much more appreciated contact, we still don't know a lot about it. And I think there's a lot of functions that are attributed to contact sites, but at this point, they haven't been definitively shown. So in this way, I think we're getting better at identifying the proteins that are involved, but what those proteins are actually facilitating, I think there's still some pretty open-ended questions.
Very nice, thank you. Yuan, so I was thinking as you were presenting your workflow, how things like oxidation that are so randomic, how the variability that these different processes introduce is likely changing the molecules affect the ability to look at them. Do you need to have a super homogeneous molecular structure, or cryo-EM accommodates differences like process modifications and slight changes in structure? Can you resolve those different structures with cryo-EM? What's your limit?
Yes, actually, I would argue that that's instead of a weakness, it's an advantage of cryo-EM, because without the need to crystallizing your target, you will be able to computationally partition your particles into different bins, and to be able to actually get you kind of a dynamic view of your system. So, and with the benefit of high resolution, as well as this a unique way to deal with a heterogeneous sample. As a matter of fact, a lot of the examples that I presented today were not recombinantly expressed proteins. They are actually directly pull it out from the cells.
Oh, wow.
Yeah.
Thank you. And then I would like to know if the mislocalization of SAMP-A, how that affects the higher order chromatin structure. Do you know if it opens chromatin, closes chromatin? You know, the reason why I'm asking is that I'm very interested in using the technology, well, you know, oxidation of 3.1 to open chromatin, and then blasting DNA to cause damage and kill cancer cells. So I'm thinking about, does this mislocalization of SAMP-A affects higher order chromatin structure?
Yeah, you know, that's what we'd like to know. I mean, so we're making the cell lines right now in order to be able to look at CTCF localization and ask, you know, since we see the co-localization of SAMP-A at CTCF sites, the first question is, does CTCF overexpression block CTCF, or sorry, does SAMP-A overexpression block CTCF localization or vice versa? And ultimately, right, we'll go in and do the higher order chromatin structure experiments, but we don't know that yet. I think you know, but the mechanism that we think it's regulating, or that it's, you know, that it's using now is through CTCF localization. I think whether that's, you know, whether that's unique to SAMP-A, or whether that is, you know, has you know, whether there are other things that are going to perturb CTCF, or whether there's other ways to perturb the, you know, the 3D organization, I think is a question, but that's kind of where we're focusing.
Thank you. And I think that for John, and I would like you to be here, you know, talking about those modifications of proteins that are not your diethyl formation. I'm totally with you on that, but I think that the sulfenic modification, the SOH, before it becomes glutathione, might be important. I don't know what are the limitations of mass spec into detecting, you know, the derivatized SOH. Is a very unstable modification. But for those of us that are at Northwestern longer, that doesn't include me, of course, I'm one of the recent arrivals. Do we have the ability to make new probes and a chemistry department that can help us to come up with new sulfonyl probes? John, I think you're muted.
You know, I'm at a Gordon conference. It's the last night, so there are drunks everywhere. So,
you mean jealous in Italy with your glass of wine?
Not me.
There you go.
Not me. But so it's very clear that there are oxidation modifications on proteins inside the cell. In fact, this came up several times. I'm at a biochemistry, molecular biology Gordon conference on chromosome organization. And in fact, a few times during this conference, cysteine modifications in vivo have played a role in the research that people have been talking about. So I think the old picture that everything is reduced inside the cell is kind of like, you know, that's something that's pretty dated. So I think a thing that can be done with the resources we have is some kind of, to start with, you know, systematic categorization of what these modifications are. And I think with some thought and with, you know, our proteomics center, it should be possible to come up with schemes for identifying, you know, where there are SOHs, where there are glutathiones, and how this, you know, where there are disulfides. Actually, I was hearing today about a transcription factor which actually does develop a disulfide in response to redox stress inside the cell, and then that
really? Interesting.
sensor for redox stress. And you know, I think determining what these modifications are and determining what they are through the cell cycle, that in itself would be an excellent starting point. It doesn't seem to be something that people have systematically done in the literature. So, you know, just determining what those modifications are And this idea, I showed you guys a slide of these papers. People talk about there being a redox. Maybe Marcelo, you're familiar with people talking about there being a redox cycle with the cell cycle.
Right.
To what extent is this true? Like, what is the redox potential as a function of cell cycle, really? And can measurements be done which are convincing? You know, can we do a study that's convincing of how that works? And, you know, for Laura, you know, how are the mitochondria, which are reorganized during the cell cycle? I mean, obviously, there's a possibility there for release of a lot of reactive species during the cell cycle, so especially in things like human cells, normal cells, and in cancer cells, where you have a lot of potential rupture of mitochondria. So I just don't know. I haven't seen papers which are very convincing about the science underlying this. I think there's a lot of room there. We actually had a symposium that was kind of focused on this a couple of years ago, associated with the PSOC. So we had a bunch of speakers in, but you know, it all seemed very preliminary and on very weak ground, this kind of biology. So I think there's a lot there that needs to be done.
That's right. Yeah, we are trying to do some of that. Of course, that everything goes much slower than we would like. But what we have been using to measure this redox potentials in organelles, and there are limitations, is this construct. I didn't talk much about that, but since we are at it, so what it is just a ORP1 protein that also is a yeast protein. It's very sensitive to oxidation. And, you know, it has just two forms that respond to different wavelengths, whether it's oxidized or reduced. And we are trying to couple that with Fushi, which is a cell cycle indicator, but generating, well, the traditional Fushi is green fluorescent protein, which obviously we cannot use with this. It's also a green fluorescent protein. But we're trying to modify Fushi and see if we can have redox readouts at different phases of the cell cycle. Fushi is also not amazing because it uses two proteins that are in G1 and, SG2M, so we don't have that much of a resolution. But at least we can see if we have redox variations in the nucleus using this construct as cells advance from G0 to G1 to SG2M. Yeah. So I think that would be a way to get at least some preliminary data to show that there is a redox.
Yeah. I mean, I don't know if you have the same impression of the literature as I do, but what's in the literature very weak, very weak.
Very weak. People have talked about that, but I think that is convincing evidence is lacking. And just so the group knows, we not only have this redox sensor, but we also have it in mitochondrial form, nuclear form, cytosolic form, and I have the animals. I have the animal that expresses the cyto, the mito, and the nuclear. So there's even mice that we can use to study this if anybody gets interested. So these are available now. Oh, there are messages on the chat, and I haven't seen them.
Marcelo, do you, in your sensors, even independent of the Fuji, right, you can probably tell whether there is a significant cycling in the population just by live cell imaging. And of course, you can for sure tell if there's a change in mitosis, right?
I'll tell you that to this group, because there's something I've observed that I don't understand. If you just make a movie of the cells and you follow them as the cell cycle progresses, and I'm not a cell cycle guy, so probably, you guys will know what it means much better than I. What we see is a tremendous flash of oxidation once the cells divide. It's almost like this flash happens to tell the cells, well, now it's time to end the cell cycle and quiesce or enter G0. But I think that the moment where we see this most clear is after the cells divide. There is a very obvious flash we can see with the sensor. We didn't explore that. Another interesting thing that we saw by coupling the sensors with the ability to generate hydrogen peroxide in the nucleus is that if you generate hydrogen peroxide at levels that don't damage DNA, you don't have a halt in the cell cycle. You actually have an acceleration of cell cycle. And I never understood that also. We expected that you produce hydrogen peroxide in the nucleus, you would halt the cell cycle. That's at least what people believe. But unless if you have that probably happens when you have DNA damage. But what we've seen at sub-damage levels, which was very, very interesting, is a tremendous acceleration. of the speed of cell cycle. Cells don't even stay in G0. They just go from division to G1 again and restart, which is very crazy. We studied that for a while. It seems that hydrogen peroxide destabilizes RB, so the cells really don't exit the cell cycle; they just enter into a new cell cycle. I never understood that then until I saw a talk by Genevieve Almusni. Very interesting. So she's proposing that boundaries of 3.1 and 3.3 histones define origins of replication of the mammalian DNA, right? Unlike bacteria, they don't have an origin of replication, but she's proposing now that these boundaries between regions that are indexed by these different variants function as origins of replication. So what I started to think, and I don't have any data to support this, it's just blabbing, is that, by messing up with 3.1 distribution, you open more sites now where 3.3 and 3.1 make boundaries. So you just have many more of these origins of replications happening under conditions of light oxidative stress in the nucleus, which is where cancer cells live, right? Light oxidative stress makes proliferation goes forward, makes them very active. So I think that one thing that I never could measure, because I don't know how really, it's just some thought in my mind, is that when you remove some of the 3.1, those gaps are filled by 3.3. So statistically, you're just increasing the number of boundaries and the numbers of origins of replication that now promotes this acceleration in proliferation. But I never measured.
So you'd assume that the S phase is shorter because you're activating all the
I know
origins
that
earlier. And that's that's okay.
I know that. So we have the movies, and we can see that if we produce very low amounts of reactive oxygen species in the nucleus with the DAO system, you accelerate the cycle tremendously, and all the phases are basically a lot shorter. G0 disappears. And if we just use the NLS sensor to look at the reactive oxygen species in the nucleus, it's difficult to see much of a difference. But once the cells divide, there is this flash of Ross that I never understood. It's super curious from an observational, but I don't know what it means. So just to make a point to what John was discussing, I think that there is something there. We just don't know what the heck that is.
Documenting that generally, like not in one not in one sick cancer cell line, but in some, across some panel of reasonable cells, I think that would be a major step forward in that field.
Yeah, I think that the only thing we don't know is what it means, but it, and you're right.
Documenting it is the first observation is the first step.
Sure, I think that we can do. We have.
So what do you use? You use a redox sensor, basically?
Sorry, John?
You use a fluorescent redox sensor?
So that's the structure of the redox sensor. It's a conjugated of two proteins. One is the typical RO-GFP. That's what Postmacher has developed, right? But the problem with RO-GFP is that it's non-fluorescent, and when it's oxidized, it becomes fluorescent. So the problem is that you don't know if you don't have fluorescence because it's reduced or not expressed. In this system, what you do is that you have a super sensitive redox protein that changes conformation of the entire sensor. So you go from having an absorption maxima at 488 to 405 when it's oxidized. The fluorescence is always in 530. So what you do is that you record the fluorescence in response to these two excitation wavelengths. You mathematically convert that to pixels, and then you express it in heat maps. That's what we do, right? And we send it to different places. There are caveats. It sounds a lot easier than what it is to use this, but we have been very lucky in getting a lot of good data with it.
Yeah. Okay. Well, it sounds interesting.
I did want to point out there is a question from a trainee in the Q&A.
Thanks, Laura. I was just going to do the same.
Let me see what it says. So Sheila's just saying that there is a dinner after this.
No,
there's
an actual question. Can you read a lot of?
Yeah. And so this is from Carla Medina. So how would one begin to think of therapeutics that can target or reduce the redox problem from contributing to chromosomal abnormalities? And so Carla's a second year in the Bachman lab, and she's curious about different classes of drugs, but she's having trouble thinking about how the issue could be translated into medicine.
Oh, there are 2 chat boxes.
Yeah.
Yeah, fortunately, I never worry about translation. I'm only interested in fundamental cell biology.
So I probably can take that one. So I believe that first we need to know what are the most vulnerable components of either chromatin or writers, erasers, epigenetic regulators, transcription factors that are vulnerable to oxidation. I think that's, like John said, it's an area that is in development. It's largely unexplored. Once we know that, we're going to come up with targets, right? I think that we also need biomarkers, and that's where the metabolism enters in a work like what Laura is doing. Maybe the positioning of mitochondria can tell us whether you have a more intense oxidative environment being created or a less intense. Of course, if reactive oxygen species have to cross the entire cytosol to get to the nucleus, that will be diluted if mitochondria is organizing right around the nucleus, making channels where oxidants can be infused. All of that, measuring the reactive oxygen species in the nucleus, establishing a pecking order of what gets oxidized, what impacts it has on function. And then, beautiful structural work like what Dan and Yuan are doing to let us know what to target and how to target. I think that it's a multiple component answer. I think there is an entire field that is developing, and that's why I'm so excited about it. I think that opportunities will come as we figure out some of the basic questions in, redox epigenetics, redox chromatin organization, and things like that.
Guys, you know what? It's nearly midnight here, and I have to get on that early bus tomorrow. So
go for it. Thank you so much.
I'm going to sign off.
Thank
you guys talk. You guys should talk for like 2.5 more hours.
Looking forward to have you back and meet you in person.
All right. See you later.
Thanks, John. Bye-bye.
Bye-bye.
And Marcelo, I do have a question about the flash in the nucleus. Have you looked at the sensor in other organelles? Is this nuclear specific? Okay.
No. The only way we used the mito is because we also have a system where we transform MCF10A cells that are non-tumorigenic. They are of course, not metastatic because they're non-tumorigenic. And we can transform them to a phenotype that is extremely tumorigenic and metastatic by inducing the expression of the VSARC oncogene. That happens over the period of about 7 to 10 days. You get a very metastatic, nasty phenotype. And we saw that the transformation to this nasty phenotype accompanies an increase in the redox state of the nucleus. We wanted to know where they were coming from. So we actually use the cells that express the mito-ORP. We see that with transformation, there is an increase in the reactive oxygen species production by mitochondria. You see that reflecting it to an increase in the reactive oxygen species in the nucleus. But if we express mitochondrial target catalase, you wipe out reactive oxygen species in both compartments. So in that case of VSARC-induced transformation of that cell line, reactive oxygen species from the mitochondria are ending up in the nucleus. And we also know that that's what's driving H3.1 oxidation and loss and everything. So that's where we started thinking about this metabolism-driven epigenetic reprogramming at the structural level, because now mitochondria can become the source of oxidants that will target certain components of chromatin. One thing that I didn't tell you guys, but I think it's very exciting, the residue that is oxidized is cysteine 96. It is a residue that is at the core of the nucleosome. It's the boundary between H3 and H4. So it's a residue that will never be modified by enzymes. It's not accessible to enzymes. It's not even close to the tail or to the surface of the nucleosome. So the only way you can get to that core region of the nucleosome is with these molecules like H2O2 that can be water solvent, but also do well at the nonpolar core of proteins like this boundary. So I think it's an evolutionary argument for why that redox relay appeared. Interestingly, we kind of did a very poor, and I'm branching to things that I'm really not versed in, but we did try to do an evolutionary study, and it seems that H3.1 appeared with placental mammals. So we started thinking that maybe that's an integration between metabolism, oxygenation, and the need of the trophoblast to become more mesenchymal and invade placenta to create the placenta. If you look at marsupials, it's not a cysteine in that position. So, and of course, cancers learn how to use all of these evolutionary things for its advantage. So we start thinking that maybe it's a way to adapt to chemo, adapt to mitochondrial dysfunction, and all of these treatments cause. And I think that one thing that I'm particularly excited with is that we could model chemo-resistant development in a dish with this. You see that chemotherapy eliminates 80% of the cancer cells, 90% of the cancer cells, but the residual ones seem to explore this mechanism to learn how to survive in chemotherapy. But if you wipe out that mechanism, then they are sensitive again. So
it might be interesting. Yeah, it might be interesting to see how accessible that site is, right? Because I think one possibility, right, is that the oxidative forms do have access. I know people have done deuterium exchange on nucleosomes, right, and looked at how fast things get protonated on the in different parts. Maybe that one, maybe that site is a little bit more accessible, but a lot of the nucleosomes they don't get whole lot of access, at least under steady state conditions, right? But that doesn't mean that there's not some other factors that could contribute to access to it.
Right. And we don't even know if, this, I think 3.1 is not going to be recognized by any reader or anything, because what we see is that it's degraded real fast. I think that's a signal of eviction, right? But we don't know if the eviction happens just because the whole nucleosome is destabilized, or if it happens because there's a chaperone that says, oh, oxidized histone, let's remove this from here. We don't know how it happens. I am favoring at this point, because of the location where it is in the nucleosome, that the introduction of an OH group there, very charged, kind of flips things around, and that dissolves the nucleosome. We are going to figure that out with you and Alex with the BioD experiments. But again, it's one example of one histone. As you know, we have discussed this in the past. SAMP A is the other one that has a very interesting cysteine. It's a cysteine flanked by two different negatively charged residues that would make that thiol very reactive to hydrogen peroxide. And there is another family of histones that have cysteines. It's actually a very rare amino acid in histones, that is the linker histones. But those are a different class. They do different things. They package nucleosomes together. That's my understanding. So there are some linker histones that also have cysteines. Other than that, H4 doesn't have it. H2 do not have it, at least the variants that I studied, AB, 2AX, etc. Among the histone trees, H3.1 is the only one. And I think histone 1.5 is the one that has cysteines. Many of these proteins have methionines, which are also oxidizable, and they're interesting, the way that they are oxidized. Methionines are even more oxidizable than cysteines. And why don't we study them? Because there's no antibodies, it's super reversible. And if you remove the protein from the milieu where it's at, just the isolation introduces oxidation. So I talked to Neil about studying methionine oxidation and histones, like his hair went like this. Right? So I think we don't have the means now to study that. But
most of the methionines are also removed from the histones. So
I didn't know that. So
yeah, so it's not, but it's it's interesting that you're limiting the seems like you're limiting the number of potential sites of.
And there are other things, like there are nitrations of tyrosines that are, there are many, in my world, there are many potential interesting modifications, some more interesting than others. But as John said, it's so unexplored, and thiols are recognizable redox switches that if we can come with a couple that have any biological function, or impact on nucleosome structure, how they interact with complexes, and Yuan would be the person to look at those. I think that those would be quite a big deal because it's largely unknown. And not to mention that it makes great use of the expertise from here and Evanston. I think it brings the two places. So I'm really looking forward to see where this takes us, but it's interesting.
Really.
So I think Marcelo, we're about ready to wrap up. It's 2 after 5.
Yes.
I just want to thank all of you. This has been amazing. I'm looking forward to continuing the conversations over dinner at Maggiano's on Clark Street in Chicago at 7:30 this evening. I advise everyone who's coming, if you're coming from the north side, to get off Lakeshore earlier rather than later. Get off like around North Avenue because it's backed up all the way through the loop thanks to all the lane closures with NASCAR. Making our lives miserable. So I'll look forward to seeing you all then, and we can echo John Marco's wine drinking.
Thank you so much to all for the presentations. Thank you for coming. It was very exciting. Thank you, Sheila, and thank you, Lisa, for putting together. Really appreciate you guys.
Thank you so much.
Thank you, everyone.
Thank you.
Bye-bye.
Thanks.
Bye.